Hello everyone, my name is Vicky Kumar and I am doing MTech in Agricultural Biotechnology from Agriculture and Food Engineering Department of IIT Kharagpur. So today I am going to teach you the most important topic of meat, that is RNA interference. RNAi. This is the part of the biotechnological application chapter of the meat, and RNA interference is basically the defense mechanism in eukaryotes. And it is a double stranded. RNA molecule which can be exogenous, which is called siRNA, which come from outside, like from viral infection. And the another one is internal, that is called miRNA. In the syllabus of the unit, the most important thing is the application of the RNAi. So this double-stranded RNA, which is the complementary. Complementary means it's chemically complementary. That means their sequences are like that, they bind to each other in 5' to 3' and 3' to 5' orientation. That 5' to 3' and 3' to 5'. So it is complementary to the miRNA of the target, complementary to the targeted miRNA. So what happens? Chop it off in a small pieces and silence it, and there is no translation, and no protein, and no action, of the protein. So what happens, in the application of the biotechnology, very classical example, that tobacco plants the nematode called, so in tobacco what happens, the nematode called melitozyme incognita, so they form the knot in the root and it reduces its production. So what we do, we genetically engineered it by using the miRNA and produce the miRNA which eventually silence the effect of the toxins done by the this nematode. So let's see the mechanism how it does. So let's now see the mechanism. is how it does, like I have said previously, like exogenous, the double-stranded source of exogenous is called siRNA, and the endogenous source is called miRNA. So now we will see siRNA mechanism. So in siRNA, the exogenous source of DNA, for example, by the viral infection, by the double-stranded RNA viruses. So what happens when it infects the eukaryotic cell? It injects its double-stranded RNA in it. So let's zoom it out and see. So when this double-stranded RNA is recognized by the system as a foreign, and it recognizes the unusual double-stranded RNA by the protein called capital always. So it is recognized by the dicer, and dicer binds at the 5' of the sense stand, 5' of the antisense stand, and it drops it off into the small RNA molecule that is called siRNA. So it chops, the dicer chops up it into a smaller fragment called the siRNA, and it also attaches some signal at the end, both the ends, and which is recognized by the
Start.
And the signals attached by the dicers are then recognized by a protein called RNAi. Which is also known as silencer. So what it does, it recognizes the signal and it binds to the siRNA, and it chops off the sensor strand and into the smaller fragment, and leaves only the antisense strand. Now this antisense strand binds to the target RNA, and and then it is chopped off in a smaller fragment by the opponent, and finally silenced and no product is formed. Now let's see the second mechanism, which is by the endogenous double-stranded RNA, which is called as RNAi. So here what happens? This was stage inside the cell by mechanism like transposons or the genetically modified it and added to the cells material, that is chromosome. So what happens from here? It transcribed into the RNA, which is then processed. So let's learn the second mechanism, which I earlier told you, the endogenous RNA interference, which is called miRNA. So in miRNA, the source of the double-stranded RNA stays inside the cell within the chromosome. And I will have to draw the nucleus bit bigger, and cytoplasm will also be bigger. So this it gets transcribed as a double-stranded miRNA, premature miRNA. This forms here because of the palindromic sequence, which we design or it naturally forms as a transposons by transposon mechanism. And
Start.
so this miRNA is matured by a protein called DOSA. So it removes this part and make it pre-miRNA. After that, it come out from the nucleus through nuclear port, and here it is recognized by the same dicer protein here. And it makes it chops off this part and make it mature miRNA, which then works similar to this, exactly like this function, and it carbonate processes it and silences the gene. So now we have learned the mechanism how miRNA silences the gene. Now we will know, as we talked earlier, that how it will be work in real life. So like this miRNA, like previously we saw in miRNA as exogenous source, endogenous source, it transcribes the pre-miRNA, which is converted to finally to miRNA, and then silences our gene. So here what we do, that in a plate we transforms our gene of interest in a agrobacterium bacterium. So now our gene of interest is in the agrobacterium, which is then infected and transformed into the our agrobacterium plant. Transformed into the agrobacterium plant, where it produces the miRNA, or in proton we can say RNAi. This interferes in mechanism of the pneumatoid here, and in the other cases, in pest control or any disease resistance, making disease resistance, we can use this. And for today, this is all, and thank you.